Assalamualaikum friends, welcome back to my YouTube channel. Hope so you are all doing well. Friends, this is my second video on my YouTube channel, and in this video we are discussing our second lecture, that is production of penicillin by fermentation. How penicillin is formed by fermentation we are all discussed in this lecture, in this video. Penicillium chrysogenum, you can say that. First of all, introduction. Also known as Penicillium notatum, it is common in temperate and subtropical regions and can be found on salted food products, but it is mostly found in indoor environments, especially in damp or water damaged buildings. It is the source of several B-lactam antibiotics, most significantly penicillin, which inhibits the biosynthesis of bacterial cell walls, affecting lysis of the cell. Next, you can see that the cell structure. Penicillium chrysogenum exhibits typical eukaryotic cell structure. It has a tubular cytoskeleton which is used for motility. You can see in the diagram structure of P chrysogenum and TAM structure of P chrysogenum. How the penicillin can be produced by fermentation. You can see that production of penicillin. P. chrysogenum is highly yielding strain and therefore most widely used as production strain in culm preparation. Purpose is to develop a pure inoculum in an adequate amount. To do so, various sequential steps are necessary, like a starter culture is needed for inoculation. After getting growth on solid media, one or two growth stages should allowed in shaken flask cultures to create a suspension which can be transferred to seed tanks for further growth. After about 24 hours, the content of the seed tanks is transferred to the primary fermentation tanks. All the bioparameters like temperature, pH, aeration, agitation, etc. should be properly maintained. pH should be near 6.5 and temperature should be 26 to 28. Aeration, a continuous stream of sterilized air is pumped into it. Bioparameters: agitation, have baffles which allow constant agitation 200 rpm. Next, you can see nutrient media. Fermentation bath contains all the necessary elements required for the proliferation of microorganisms. Generally, it contains a carbon source, nitrogen source, mineral source, precursors, and anti-foam agents. First of all, carbon source. Carbon source is lactose in a concentration of 6%. Other carbohydrates like glucose, sucrose complex, as well as cheap sources like molasses or soya meal can also be used, which are made up of lactose and glucose sugars. Next, you can see nitrogen source. Nitrogen source. Ammonium salts such as ammonium sulfate, ammonium acetate, ammonium lactate, or ammonia gas are used for this reason. Sometimes corn steep liquid may be used. Mineral source. These elements include phosphorus, sulfur, magnesium, iron, and copper, which generally added in the form of water soluble salt. Precursors. Various types of precursors are added into the production medium to produce specific type of penicillin. For example, if phenylacetic acid is provided, then only penicillium G will be produced, but if hydroxyl phenylacetate is provided, then penicillium X will be produced. Phenoxyacetic acid provides precursors for penicillium V production. When corn steep liquor is provided as nitrogen source, it also provides phenylacetic acid derivatives. Therefore, it is widely used in the production of penicillin G. Anti-foam agents. Anti-foaming agents such as lard oil, octar, decanol, and silicones are used to prevent foaming during fermentation. Recovery. The recovery of penicillin is carried out in three successive stages: removal of mycelium, countercurrent solvent, extraction of penicillium, and treatment of crude extracts. At the harvest, fermentation bath is filtered on a rotatory vacuum filter to remove the mycelium and other solids. Phosphoric or sulfuric acid are added to lower the pH 2 to 2.5 in order to transfer the penicillium to the anionic form. Then the broth is directly extracted in a Pobiolanic counter-current solvent extracted with an organic solvent such as methyl isobutyl ketone, amyl acetate, or butyl acetate. Penicillin is then again extracted into water from the organic solvent by adding an adequate amount of potassium or sodium hydroxide to form a salt of penicillium. The resulting aqueous solution is again acidified and re-extracted with methyl isobutyl. This is all about the second lecture, that is production of penicillin by fermentation. Inshallah, we will meet in the next lecture, next video. In the end, I request you to subscribe my channel, like my video. Thank you so much.